So we had started to talk about RNA sequencing in the last class, and we've been covering it for a couple of last classes right now. And one of the changes that I want to do in the course post midsem is just spend more time between theory and data. So we'll keep switching between the context, right? So that you have context, and it doesn't have to be isolated session. So for this session also. For things that I would like to motivate, I will switch between the hands-on. It's not a new hands-on, but we'll continue to work with the same hands-on that we had last time. And I'll tell you about like what is the problem with working with this kind of data. It's why is it probably not the most straightforward data to analyze with, how to think about it from first principles. Maybe. Thank you. Before I get started, any questions about anything that we discussed last time? EM is clear. So where we ended last time was we were able to quantify it. We were able to quantify it at the isoform level. And today I'll show you how I collapse this at the gene level. But overall, what our goal for today is once you have this data quantified across cases and controls, and we will see this in just a bit, how do you actually analyze it, right? So what you're seeing here on this particular plot is, in this particular figure is two examples. One of them red and one of them green, broadly, which means these are two different conditions. And when you align the data, it might look like this, right? And the simplest thought that you would probably have is you have more reads which are mapping to condition A as compared to condition B, or condition red versus condition green. Then that would imply that more number of protein or more number of mRNA copies will come from this particular sample, right? This is, in other words, more present as compared to the green one. The goal for today is to understand why that simplistic assumption is often not true. And we'll do this through working with data and trying to figure out how to get rid of it. Okay. So before I actually show you the data, what is the problem? The problem broadly is, given you have a sample A, which is sample red, and sample B, which is green, you'll see quantification coming from gene X and gene Y. These are individual reads that are mapping to gene X and gene Y. These could be any two genes in both the conditions. And what I would like to compare is which genes are differentially expressed. What that term essentially means, we will see this in one by one, is which of these samples has more amount of reads coming from one gene as compared to the other gene in a statistically robust manner. So if time permits, we'll start discussing the hypothesis testing regime. Some of you are actually there in other classes, so there's some overlap. But one of the things that I want to talk about in the class is usually not talked about in the classes, which is about multiple hypothesis testing, but one step at a time. Okay. So overall, the goal here is given two samples, how do I find out which genes are differentially expressed? Differentially expressed means statistically which gene among all the 20,000 genes that you have, which are protein coding, have either higher levels or lower levels across two samples. That's basically what the idea of differential expression is. And if you think about it, like this is just a counting problem, right? You have, let's say, 10 reads mapping to gene X, 3 reads mapping to gene X in the other condition, maybe some 8 reads mapping to gene Y, and maybe again 3 reads mapping. So it should be a very straightforward thing, right? But it is not, and we will see this example in just a bit. So let's switch to the hands-on. If it's okay if you're not carrying your laptop, because again you already have access to the thing. But what I have done, just so that we are all on the same page, is I have put this entire content as a zip file. So in principle, you can download this zip file. What it has is, somebody at some point had asked me that how should you run this script if you want to do it for multiple samples. So what you can do is you can write a bash file. So these bash files are basically nothing but just those same commands that you would have otherwise written on your terminal. This is written just as you would write your Python file or your R file. You can write what's called a .sh file or a bash file, which you run in your terminal, right? So if you type bash this file name, it will run and will do the kind of steps that you've already done. So for example, let me show you. So if you download, it will be this kind of directory, right? The first thing will just download the fastQs for you. The second one is literally just downloading the FASTA, the CDNA FASTA and the GTF file. The third one is creating the reference. The fourth one is how to map the data that you got. And finally, the fifth one is the command to create this T to G file. So last time we had talked about what am I doing with my T to G file, right? So when you quantify your data from Callisto, it will quantify this at the isoform level. However, what I want to do the rest of the analysis is at the gene level, because isoforms are too many. We should count actually. Let's count how many isoforms we have. So I'll take a lot of tangents and just see how far we can go. Okay, so first thing that I would like to see is, so this is your T to G file. I can check how many overall transcripts it has. It has around 207K isoforms, right? And these isoforms could be repeated, each gene could have multiple isoforms. So I could summarize it if I want. So for each gene, the N here shows the number of isoforms it has. Of course, this is a tabular format. I could just do and most genes have, well, this is not very descriptive, but let me do this. So each minimum number of isoform for gene is 1. The first quantile is also 1. Median is also 1. Mean is 5. Third quantile is 7. And there is a gene which has 192 isoforms. Okay, let's see what that gene is. So this particular gene, again, I have quantified this at the gene level. It has 192 isoforms, and the next gene has 156 isoforms. Let's see what that gene is. So this gene is MAPK10, which has 196 isoforms or 192 isoforms, whatever that is, right? So this particular gene name is one of those genes which seems to have multiple isoforms. Now, again, having looked at this, because this is a totally unpracticed pitch, I'm now curious, what does this look like? So I will go to genome browser, which we have done. This is an exercise we have done in the past. I'm going to just go to genomes, and it will do what it is doing. And I would like to just visualize what do those isoforms look like. So anytime you're curious, right, there are a lot of tools to help you sort of satiate your curiosity. And this is one of those tools which going forward when you're doing projects or trying to sort of get deeper will be a good resource to sort of keep handy. Right? So now I'm very curious about that gene. So what I will do is I will just go and type there, and that's the one which we want to find out. And you can see each of this here that you're seeing is one individual isoform, right? So you can see what's going on here, right? There is, I can zoom in a bit. So this is the gene. Whenever you see this kind of rectangle bound, that's an exon. You can see it's an extremely long gene. This is, I think, 34 kb. Wow, that's pretty long. 1 second. So this shows you the, wow, it's pretty long actually. Okay, so this is 100 kb region. This is around 300 kb gene actually. This is what it shows you here, right? Overall length of the gene is around 344 kb. Longer genes by definition means they usually will have longer introns. So what you're seeing here, for example, these are introns. These small things, very, very small things that you see, those are exons, right? And that's what essentially are making the different isoforms. It's a long gene, usually means more number of exons, usually means more number of possible combinations that you can have, and that explains why this has 192 isoforms, right? So never settle down for the observation that you make, just understand why it is like that. Okay, that was a good tangent, but let's go back to the overall study, right? So what we had done is at this step, we had quantified the abundance, right? So if you go back and read your T2G file, this is what it would look like. That's what I showed you. We ran Callisto. Callisto outputs a TSV file, which is called abundance.tsv, which is what I'm loading here. And what it shows you is this is the target ID, which is the transcript ID. What is the length of that isoform? What is the effective length? So the effective length is overall a measure of how much coverage does that transcript have in your data, right? So the length is a function of what is there in your GTF file. The effective length is something that you notice based on your data, given what is your read length to begin with, because that will determine how much read can you cover overall and how much coverage of the read length that you see, right? So you could have a gene which is this long. So this is your length, but it's possible that it could only be covered till this end for whatever reason, right? So that in a sense becomes a measure of your effective length. And this is a function of how much uniform coverage is there in your data, what is your read length, and so on. The final column shows you, the third column shows you estimated counts, which is how many number of isoforms after we ran that EM step, expectation maximization step, we assign the reads to isoform. So let's say this is isoform 1, this is isoform 2. The third column is just showing you how many number of isoforms, how many number of reads map to that particular isoform. Okay, clear? So I could have worked with this data, but it's too much to deal with, and you can always come back to this analysis. But it's a pretty large data set, right? Again, 207K transcripts, very hard to sort of do a first pass analysis. First pass analysis should be very, very simple. To do that first pass analysis, what I'm going to do is I'm going to collapse this to the gene level. The idea behind collapsing this to the gene level is to just say that you will sum up the isoforms of each of these reads, each of these genes, and make one count out of them, right? So if this isoform level, let's say isoform 1 belongs to gene 1, isoform 2 belongs to gene 1, and isoform 3 belongs to gene 2. The counts that you got are 10 and 5 and let's say 1. At the gene level, I can summarize this by saying this is the counts that you got at the gene level. Okay, 15 and 1. Very simple, just arithmetic, just simple sum arithmetic, nothing very complicated. Yes, so it is counting the number of reads that got assigned to the isoform. That's what your third column is, right? So this has 2 isoforms, but the overall count for that particular gene is 15. the number of reads that got mapped to gene 1. Clear? Ujiti, clear? Clear? Good. Fine. So, but I also have this data, by the way, right? Like I don't have to do this step because when I downloaded this from Geo, I do have this data quantified anyway. 1 second, I need to go back to my Geo counts. So if you remember, we had downloaded this data from Geo as well, where they indeed have already quantified this at the gene level, right? So if I take one of these files randomly and I look at this is GSM whatever 39 whatever 8232, then they have deposited this data already. They have deposited that for this gene, these are the number of counts that we see. I don't remember actually which method they used, but it doesn't matter. So in most cases, this kind of data you will already have access to, which means that you don't need to explicitly quantify using Callisto or any other tool. The only reason you would want to do this is because, let's say you are analyzing different data sets across different papers, and you want to make sure that there is no systematic difference in how their data was quantified. So you would want to run the same tool on all the data sets so that they are quantified in the same fashion overall. Is that clear? So we already have this, right? You had downloaded this from the Geo website. Everyone remembers this, right? We downloaded this from the Geo website. This is what it looks like. Now my next goal is to first make sure that even if I did quantify, am I doing something which is very random, or is it something which I'm able to recapitulate, I'm able to sort of reproduce at my end. So while Callisto quantified this at the isoform level, I am going to collapse this at the gene count level, right? So this is what this level of code does. So what it does is it basically sums up it creates a column called Callisto counts by summing up the estimated counts column after grouping them by the gene ID. Is that clear? So you're taking the gene ID, right, from your Callisto quantification. You're summing up everything, all the counts that have the same gene ID, right? Very simple arithmetic, right? Any questions about this? The code looks complicated, but that's the essentially step it is doing, right? So the second step that it is doing is I'm joining two data frames. I'm joining the data frame of the Callisto counts matrix with the T2G, because in the Callisto counts matrix, I have the transcript ID, but I don't have their gene ID, which is what I'm getting from this column. Okay? That's what this particular code block is doing. Finally, I am removing, if you see the T2G column, the T2G data frame, then it has these weird numbers at the end, which I don't want, because usually they're not very informative, they're not required, and also my Geo data set does not have those numbers. So if I want to compare my Callisto quantification to my Geo quantification, I'm just getting rid of this in that particular column where I'm stripping the dots. Okay, this is what this particular code does. It removes dot one. So the comments are there. I don't have to necessarily explain each of them, but essentially you are summing up the counts overall. Okay. Okay. The reason I'm doing this is because I want to make sure that they correspond to each other, right? That even if I were to do this analysis on my own, I will be able to reproduce my analysis. And this is the plot which shows that when I quantify using Callisto and when I compare this against the same quantification that they have done at the gene level, it kind of corresponds, right? It's not fully exact, right? It's not like a straight line. If everything corresponded 1-to-1, you would get a pretty tight straight line, which doesn't happen, which can happen for multiple reasons. It can happen because, first of all, the quantification strategy is different. It could happen because the way I'm summing up at the isoform level, the GTF file that I use maybe has more isoforms, less isoforms, multiple possibilities exist. For this class, it doesn't matter. We don't have to get into the details. And they're not really required because honestly, there's nothing more that you can do to fix this, right? Because the data set already exists. You should ideally go ahead with your quantification in this case. This just provides you a view because why? Because you have the isoform level quantification. So you can always come back and do any analysis at the isoform level, which you would not be able to do with your gene count level data, okay, with your geo counts. Sure. So what I'm plotting on the x-axis is the total counts at the gene level, which I got from my quantification, which I did using Callisto. Okay. On the y-axis, what I'm plotting is what I got for the same data set, but from geo, which was deposited. I'm trying to make sure that my quantification strategy is not extremely different from what they used, right? Which basically means that I'm able to reproduce to some extent, right? To some extent, you're able to reproduce this analysis that they did, because you see a very tight line. It's an x equal to y line, by the way, which in R is very, very easy to plot, and I think it's easier to plot in Python as well. You just do geom AB line. This is your x equal to y line. So if everything corresponded, this should have looked very straight. It doesn't. You can calculate the correlation, for example, that if you want, let's do that. It's not explicitly done here, but I can just do we'll have to do remove. So 0.97 correlation, right? It's not bad at all overall. It's okay overall. Does that make sense? So what I did using Callisto at the gene level is what corresponds to what they deposited out there. In an ideal world, you will quantify all your data using Callisto or any other tool that you like, and keep proceeding. We cannot do that because there are 196 files that we will have to do this for. So what I am, okay, I do show the correlation here. Okay, so ideally I would want to repeat this Callisto again and on all the 196 files or however number of data sets are there, but we don't have time for running that in the class. So what I'm going to do for the rest of the class is just use their deposited data for doing the analysis. Okay. So to do that, I have, well, okay. That's where the issue is. Just give me 1 second. Sorry, I forgot to put this file. Just give me 1 second. In the meantime, I'm happy to take any questions or any other things about projects while I'm looking for that file. Any questions? No questions? Okay, I'm not able to find it, so it's okay. We're going to recreate it from scratch. It's not a big deal actually. So the issue is that when we load one of those files, so this is what your file looks like, right? It does has 2 columns, and ideally what I want access to is this should be a long data frame which involves your gene IDs, and then for all other samples, for all the other 196 samples, this should be a full-on matrix rather than just one matrix. Okay, so we'll try to do this, and we will run into issues, but let's see, we can do this. Okay, so Okay, so maybe I think it's a good idea for me to also walk you through how to do this actually, rather than actually do this. So first thing that I have to do is I am going to iterate through all over the files in this folder, because all these files are in this folder, right? So the way to do this is I'm going to say that if you do just list dot files, it will list all the files which have a particular extension. So sorry, bulk, not this. Okay, so this is the list of those files which exist in my directory. So I have all these files available. I will have to write a custom function to do this, and I think that's why I had prepared that file. But I am going to do it right now, right? So maybe you want to walk me through the logic that you will use to read all of this in a data frame. How will you do it? The goal is to create a matrix which would look like this, right? So what you want on the rows are your gene names or your gene IDs, and each sample should follow along. Okay, so there are 216 files in your directory. Each of the file has very simple format, is gene ID and the counts. From there, you want to create a data frame which looks like this. How would you do it? Any ideas? Mm-hmm. Okay, and sure, yeah, very simple, right? So that's correct. So you would do this, which is I'm going to reiterate through. Ashwa, you were saying something. Okay. Okay. So in R, let me try to see if it works. So let me not speak it before it works. But I could do this, for example. And one thing that I would like to do is I had downloaded metadata for this, right? So this is, for example, what that metadata looks like. So if you think about, sorry, this is the wrong one. So what I want is, I also want the record for each of this gene expression IDs, right? So for example, this is one of the samples that exists in your data. This comes from, see, this is not correct. Sorry. So this comes from this particular data set. What? Oh, did I step on the power supply? I think it just went actually. I'll try to find. This also went away, right? No, this is on battery. This is on battery, but it is connected to the amplifier. What did I do actually? Let me call him because I have no idea how this works. So the entire power went away. I'll have to call him. Sorry, can't find him, so have to fix it ourselves. Any ideas what went wrong actually? I have no idea because because everything is, all the switches are here, but how it happened that I don't know. All the connections are here actually. Okay, that's okay. So since we can't do this, we'll move to the board, analyzing our overall description, right? So now let me maybe talk about, you will have access to slides, so I'm going to just, because I don't want to waste time finding the person. Oh, how did it come back? Oh, okay. Thank you. Thank you for telling me. Good, good time. Hmm. Hmm. No, it will not, because by the time you have summarized it here, you have lost that information, that which amplification cycle it came from, and good question. Yeah, yeah, sure. So that is something that you handle at the mapping end, right? So our question is that if a gene has a lot of repeating structures, right? So maybe there are a lot of A's. Is that what you mean by repeats? And then there is a lot of T's. So if you think about it, like actually a lot of our genome is repetitive, in the sense that you will see a lot of these repeat sequences there, like almost 50% or 20, 30%. I don't remember the number right now, but it's very big overall in terms of how much repeats do we see in our data. So these regions are hard to map, right? Because if there are so many of them, how do you tell that that particular read came from here? So again, we use that EM idea to actually resolve some of it. But once we have done this, we are now at the level we are actually working with quantified matrix. So if your aligner is not able to solve it, at this point, we just move on and move at this step, right? There's nothing more beyond that that we can do. Yes, you can. Yes, you can. You can hit a jackpot because of repeats. Yes, that can happen. But in most cases, if you have repeats, right, your aligner will try your best to resolve. But it is quite possible that you don't get enough quantification if it is not able to fully resolve, right? So if it is multi-mapping, you will not get enough signal from those regions. So the way it is handled is post hoc, which means that you analyze the data first, and then you try to comprehend that did it happen because maybe there are a lot of repeats coming from that region. Okay. Okay, so this is another example on the same thing, that how can we compare RNA sequencing across two samples, which is what we are going to do in the hands-on as well. So this is different genes, and again, I think it's a similar-ish example where everything is similar, but if you focus on gene C, it saw a 10-fold amplification overall, right? Um, which could be biological or it could be technical. The challenge in analyzing any of this data is sequencing depths are different, or technical differences exist that could mask your biological differences. And there is one more problem actually, is this quantification that you see, right? Like some gene having more counts versus some gene having less counts is also a function of the length of the gene. The longer the gene, more number of isoforms you would get, because when you're sequencing, so if this is a shorter gene and if this is a longer gene, just because this is longer and I have sequenced everything uniformly, hopefully, this will get 2 reads, and since this is 2x long, you might get 4 reads, right? Because you need more reads to cover it. So ideally, you would want to essentially also control for that fact that different genes have different lengths, because that will tell you how many counts you're getting. So if it is getting small counts, does not necessarily mean that it's a lowly expressed gene. So I would like to quantify that how or what is the relative level of expression of each gene before sort of trying to do cross comparison. I want to do both. I want to compare relatively, is gene A more expressed than gene B, in a normal sample. And then I also want to compare that, okay, I know this fact that, gene A is highly expressed, then is it also differentially expressed across my conditions? Okay, is that clear? Okay, so one of the simplest ways to do this is what you suggested originally, which is I want to compare the counts. What I could do is I could sum them up and I could divide this by each of these numbers here by that full total count, right? So this sum has 577, this sums up to 460. I can divide it, and since this number is going to be very, very small, you multiply it by a large factor, usually which is 1 million or 10 to the power 6. And this normalization is called counts per million normalization, right? These are the number of counts that you have. Once you normalize it, that's what you see there. These are the number of isoforms, or these are the number of overall mRNA copies of that gene that you end up sort of observing, having accounted for the total sequencing depth overall, right? So after doing this normalization at some point, you would be able to sort of compare, well, the goal is that you should be able to compare these two quantities, right? But what is the flaw in this logic? Like why does this logic not work? Sure. So if there is an outlier, then of course, it will inflate these counts. That is number one reason. The other reason is if I want to compare one gene to the other gene, even within the same sample, right? This number actually does not do justice, because while I'm showing you the number here, 20797 and 69,324. I cannot compare gene A versus gene B even within the same condition. Why? The length of the gene is different, right? Which has not been accounted for here, right? So gene B could have been 4 times or 3 times shorter as compared to gene A. So if you do this kind of normalization, it doesn't actually help you do any sort of summarization. Like, it doesn't actually help you. You cannot compare this across samples because one gene can skew your entire analysis. You cannot use this number to even compare within the sample, because again, for the same reason that your length, well, not for the same reason, but a different reason, which is your length is not accounted for here. How will you fix this? Let's focus right now that I want to compare genes, the relative abundance of genes within the same condition. How would you fix it? Okay, good. In a stable manner. Okay, and how will that work? Wow, that's a very good idea. So you've invented a new normalization thing, which is very, very good. But how does it actually tell you, how does it account for the fact that if I take, let's say, 5 genes, right, which are otherwise expected to be, what you're calling them is they are expressed everywhere, right? Like they're expressed at the same levels. How does that help me, their levels, how does it help me compare the rest of the genes, right? So for example, let's say this gene is stable, right? 5 is the number. What do I do after that? So I can compare, you are saying that I can calculate 1 by 5, 3 by 5, this will be 1, and this will be 1 by 5. And there will be 10 by 50, 100, so 1 by 50, 35 by 50, 35 by 500, and this will be 55. Well, we chose a wrong example because it's quantified 100 times, but that's what you would like to do, right? Okay, so I'm going to still compare, if I want to compare these two, I still have that same problem, right? That this 1:3 ratio does not account for the length of the gene. This doesn't necessarily tell me that there are 3 more copies of the gene, because Is it because it is longer, or is it because it's actually biologically more expressed? Does that make sense? Yeah. So this is a hypothetical example. He said that if you know a priori that these particular genes are stable, which means that they're not variable across samples or across conditions, and there are these genes. These genes are broadly called housekeeping genes, which is they are on every time. They're there because they're keeping your cell alive, right? So these some very ubiquitous example of those genes are what's called ribosomal genes. They have to be there all the time. Like they have to be on all the time, mostly. So if you know about certain genes, you can use that as a way to normalize your factor, right? This is the same level at which they need to be expressed. And if you know that level, so that level is, let's say, 5 here, and for whatever reason that was 500 here, I could normalize everything with respect to that particular gene. Okay. Okay. So okay, fine. So let's say it's this gene length was, let's say, 5, right? So 5 by 5 and 500 by 5, right? After that, what do I do? Mm-hmm. Hmm. So getting there, but I think it requires a bit more the normalization factor, right? That normalization factor could have been if you knew the length of each gene, then what you would essentially do is, that, for example, this gene length is 1 kb. I could define a quantity which is called FPKM or RPKM. The distinction between FPKM and you colloquially, they could be called reads per kilo million, kilo base million, or fragments per kilo base million. It's called reads if it is single end, if it is called fragments if it is double pair end, but for all other reasons, they are basically practically the same. What you do is you divide the xi, which is the counts, upon the length of that gene, and you also divide it by the total number of the entire transcripts that you see. Now, in the process, hopefully, what you have done is you have accounted for different length of each gene, right? So these numbers that I'm seeing right now, these are, I'm taking the same CPM, right? So if you see, this is counts per million. This length is the same, or this is the only, sorry, this xi by n is the same as in CPM. This length is what I'm dividing each of these genes as, right? So from the previous figure, from the previous table, I'm just dividing it by the length of the gene. Now, hopefully, I can compare the gene levels across within the same condition for these two genes, right? So if I see 34,000 fragments and 207 fragments, this likely indicates that gene B is more expressed, right? Because I've already accounted for the length of that gene. It's already been regressed out. It's already been accounted for overall. So if I sequence, what is the interpretation of this, right? If I sequence 1 million fragments, yeah. Why intron length? Right. Correct. Correct. So you mean exon length, right? Yeah, sure. So that's what actually that's a very good point. This is that effective length essentially captures the length of the exons. I did not push on it, but that is exactly what you do. Yes. So rather than the gene length, it is actually the sum of the all possible exons that exist, right? That defines your entire gene. That's correct. Okay. Now, so what is the interpretation that if you sequence 1 million fragments, you will see whatever that number is, you will see that gene getting those many fragments of this particular gene will be sequenced if you will sequence 1 million fragments. after accounting for the length adjustment that you have done, right? Is that interpretation clear? So you're essentially saying that if I sequence everything to be 1 million, after having accounted for the gene length, I would, or assuming all gene lengths are the same, I would get 20,000 fragments from gene A and 34,000 from gene B and so on. Okay, is that clear? Okay, can you use this to compare two different conditions? So you have accounted for total number of reads or total number of fragments. You have accounted for gene length. Can you use this to compare one set of samples to the other one? Why? So in some sense, you've not actually accounted for that fact that, you know, can still have crazy genes, right? Like you definitely cannot use this for comparing this across conditions because there is no adjustment explicitly that you have done. The only adjustment that you have done here is accounting for the length, right? That's the only adjustment which allows you to compare this within the condition, but it doesn't allow you to compare across conditions, right? So people realized this and they started to sort of think about that how can we make this number a bit more interpretable. And one of the things that they realized is that the FPKM values, when you sum them up, they actually don't sum up to a constant number. So despite doing that normalization where you're dividing the total counts by the length and by the total number of counts, when you sum this across different samples, this number, if you sum this over your sigma xi, and this is li, this number can be very different for anyone, for any particular sample. So they said that if you want to sort of, let's think about a number which will be kind of comparable or, you know, comparable across two conditions. Let's make sure that we can account for this fact that this xi by li ratio is going to be different overall. The way they do this is by essentially taking the same quantity and dividing it by the sum of this, right? So if this number is going to be different across conditions, let me sum this number over and take the original quantity again and just divide it by this number, right? So this is normalization of the normalized value that you have. Now that interpretation becomes much more simple, right? What it essentially means is this is basically what's called transcripts per million, right? Transcripts per million. If I were to sequence 1 million fragments, then in each of these conditions, then the TPM value, whatever I have here, will be the number of fragments or the number of reads that I would get for that particular gene, having accounted for all possible things. Does that make sense? So that now once I've normalized this, right, all of them will sum up to the same number, and that number is 1 million. In the case of RPKM, when I was doing this calculation, I think I need to fix the second table. If I sum up these numbers, they will sum up to two different numbers in these two cases. Okay. But however, if I sum up these numbers, I will have to fix this table. This should show TPM and not FPKM. These will both sum up to 1 million. Does that make sense? And that in some sense makes it slightly more comprehensible that if I were to sequence 1 million fragments to both of these two conditions, these are the number of fragments that I will get from condition 1 versus condition 2. Is that comparable, however? A lot of people do this classical mistake, including people in this campus, that they compare TPM levels of two different conditions. Why is that usually a bad idea? Still doesn't account for crazy outliers, right? So you can get lucky and, you know, you can get away with by actually comparing those numbers. But if something went wrong in your sample and you compare TPM, that's incorrect. Correct. But if it is outlier in the sense that it is skewing the entire counts, it's an outlier by a factor of 10x. Since one thing which I should have highlighted before, is RNA sequencing. When you do RNA sequencing, everything is proportional. You are not sequencing the absolute RNA content. Okay, let me give you an example. And that's why that strategy, what you said, will not work. Let me give you a very quick example. So let's say you have gene 1, gene 2, and gene 3, right? Now, when you decide your sequencing depth, let's say you only have a machine which can sequence 100 fragments, right? 100 reads. So in the first run, I get 5, and I get 50, and I get 45. Since this machine can only sequence 100 reads, I could have also been able to do this. I get unlucky and I get this crazy number, right? Which is now, this is increased by a 5-fold cutoff. What went wrong, right? Either this gene hit a jackpot or this gene 3 hit a jackpot in the first case, right? It could be technical, it could be biological. At this point, I don't know. But because this went down, something has to go up because everything is proportional. Okay? So RNA sequencing is a proportional technology, right? It does not capture all the absolute RNA, because if this is your RNA, right? When you actually put, so this gene has 2 copies, this gene has 4 copies. When you put this to sequencer, and if everything is uniform, if I get 1 copy of this, very likely I will see 2 copies of this. But I will never be able to capture all the 2 copies and all the 4 copies. Everything is proportional. Sure. So let's say you have two copies of the mRNA in the actual system, and you have 4 copies. When you do RNA sequencing, when you sequence these fragments, these are sequenced in a proportional fashion, right? So everything is driven by proportion. So if I'm able to sequence only 3 reads, okay, very likely I will get maybe 1 read from here and kind of 2 reads from here, because everything is proportional. So the proportion is 1:2. That final proportion has to remain the same. I could have also, I will come to your point. I know probably what question you want to ask. I could have also sequenced 30 fragments, in which case I would probably get 10 and 20. I don't know the absolute number still. I could have over amplified them, but since everything is uniformly sequenced, right? Or in some sense, this is biased sequencing, because the initial proportion of fragments tells you which fragments are more abundant, right? So this kind of mRNA is more abundant, so it has more chance of getting sequenced. And when you actually sample on your sequencer, this has more chance of getting sequenced and less chance of, and this has lesser chance of getting sequenced. Everything scales by proportion. Sure, right? So if you are, since everything is proportional, right? These all proportions sum up to some fixed quantity in some sense, right? So that's what your TPM normalization does. When I sequence everything to 1 million fragments, then that number shows you that these are the number of fragments that I get. Now, if I run any normalization, if I run TPM, for example, if I calculate TPM, I have forced all of them to sum up to the same quantity, right? That is happening because of normalization, right? That is happening because of normalization overall. Now, because of this normalization, if something went crazy down, if this goes up, something has to come down, right? This goes down, and that's why this has to go up. This kind of remains the same. Overall, this effect cancels out. So there's an entire field called compositional data analysis, where the idea is of working with proportions. This is one thing which you should always remember, that where there are proportions, if something goes up, something has to go down, because they all sum up to the same quantity, right? Okay. So how will we fix this? The way to fix this is to work on this simple analysis, right? So hopefully you are convinced that none of these values can be used to compare abundance across conditions. How do we fix this? And it's a crazy fix which works. And again, it's one of those things which, how somebody thought about it and how it works, slightly hard to see, but when you actually see it working, you will be convinced that it actually works. Okay, so let's talk about it. So this is one which I already highlighted. So if this is a relative measurement, if one gene either goes missing or it goes ORI, then all other genes will have higher relative expression or lower relative expression, which is the example which we discussed. So we need to come up with a strategy which accounts for this kind of differential abundance structure, where we can compare samples across two or more conditions. And the way this is done is using this size factor normalization architecture, which was first proposed in 2010 by Anders and Huber, and then later on developed into a slightly more statistical argument in 2014 by Michael Love and Huber. We will cover that in just a bit. But the idea behind this strategy is to take the geometric mean for each gene, which becomes your reference. You divide the gene counts. You don't worry about the gene length, right? You don't worry about gene length because your gene length is fixed across two conditions, hopefully, right? That gene length usually will not change. You divide each gene count by their geometric mean. This is the size becomes your size factor of the sample, which is defined by median of this quantity across the sample, across all the genes within one sample. We will do an example and become clear what it is. And finally, you divide the sample by this size factor. Okay. So the first step that you do is, this is gene 1, gene 2, gene 3. A lot of samples, calculate the geometric mean. So in this case, a lot of numbers here. Sorry, so we're going to quickly take a very simple example for which we can calculate the square root. So this is sample 1, sample 2. Let me invent the example again. Okay, so the first thing that you're going to do is you're going to take the square root of 50. So for this, you're going to take the square root of this number, which is going to be pretty large. And then you're going to take the square root of this number, which is square root of 9. So this is approximately how much? Yeah, some 7, right? This is approximately how much? 50 root 10, correct, that's the right way. So 150, correct, that's correct. Okay, and how much is this one? 3. Okay, next step that you're going to do is you're going to divide this number by this number. What have you done, right? Like, what is the central premise on which it is working? This is the central premise on which it is working is that when you have a differential setting, it's not that all the genes, even if I have cancer, it's not like I will have changes in all my genes. Like, it will be a very small subset of genes, 10 genes, 15 genes, 100 genes, but you have 20,000 genes. You can always assume that at least 50% of your genes will show no change or no differential expression overall. And that's why whenever there are outliers, whenever there are sort of chances of something going wrong, your geometric mean is more stable than your arithmetic mean. We will not prove it, but there are ways to sort of intuitively see that argument, why geometric mean is a good idea. Everyone understand what is geometric mean, right? So given 3 quantities or whatever, x quantities, it's n quantities, it's just square root or whatever, cube root or 4th root or 5th root of however many numbers. That's a geometric mean. That's more stable than doing a plus b plus c. You must have seen one inequality in your 10th standard or 12th standard, which kind of actually explains why this happens in some sense. AM is greater than GM is greater than HM. Everyone has seen this? Arithmetic mean greater than geometric mean greater than harmonic mean. When you study this in 10th class, it doesn't make sense. But then when you actually come in here, it makes sense, right? So that's what a lot of our education, unfortunately, is. But anyway, so here you have divided these numbers, right? Now, in this sense, what you have done is, why are you doing this? You're doing this because you're saying that this is my reference level, right? So Aman was talking about creating a reference where you could compare this number with. By doing this normalization thing, you are essentially creating a pseudo reference for each of these genes. Given 50% of them are not going to change, you expect that this geometric mean is in some sense the average essence of how you should, what is the distribution of these genes that you should see, right? So when I divide this, essentially what I'm doing is how much is it different from that reference level. Now, what you have done at this point is just compared that how different is each individual gene in each sample as compared to a reference that you created. You do this across different genes, but when you do this, what happens is if this sample was 100 times more sequenced as compared, or overall its sequencing depth was much larger, that should reflect in some sense uniformly across all the genes, right? So if I sequence, in this case, what was my total? 55 and 58. And here it was 513, right? Sorry, it's getting a bit messy, but it is 10x higher, right? Notice that this is 10x higher in terms of sequencing depth. Calculate these numbers. So this is approximately 1, this is approximately 10. Say that again. Oh, sorry, that's correct. Sorry, that's correct. So this is 3. Sorry, I'm going to make it messy, but doesn't matter. All the slides will have it, so don't worry about it. So 1, also it's a different example, so it doesn't matter. Anyway, 1 by 3, and this is maybe, let's call it 1. Okay, 1 by 3, 1. So what is its 2.33? What is this number coming up to be? 5 point something. Somehow the effect of that depth is much lesser in this case, right? It's not as nuanced. Why? This is by design, because this gene went awry in some sense, right? Everything was skewed because of this gene. But then when you calculate the median of this number, it again, because 50% of the genes are not expected to change, this median number that you see is in some sense how overexpressed each gene is with respect to pseudo reference. That should be a reflection of how deeply sequenced is this with respect to this pseudo reference. Does that idea make sense? No. Okay. So this part clear, like why you're creating the pseudo reference? Okay. Now, when I divide this number, right, with respect to this pseudo reference, this is a measure of how much overexpressed or underexpressed is my gene. So you see a number of 1, you see 1 by 3, you see 1. Now I'm saying that 50% of the genes you should not see any change, right? Like in the sense that they're not differentially expressed. So these numbers that you're getting are a measure of how overabundant or less abundant each gene is in each sample as compared to that reference sample, right? So 1 by 3, 1, 1. Okay. And in the other case, that number is whatever, 1 point something and 1 point this, whatever, 1.33 and 3. Okay. Now 50% of these genes should not have shown any kind of change in terms of their overall abundance, right? So imagine if no change had happened, right? If no change had happened, then all these numbers would be identical, right? Because of course there's no change that has happened. In this case, something did change. What I'm going to say is that since 50% of the genes which are going to be here, right? This is the median, so this is the middlemost value. There's no change there, right? So if you take the middle value, then that essentially is a factor of telling you that how different is this sample from a pseudo reference sample. And that is a measure of overall my entire gene signature, my entire set of genes are by how much deeply sequenced or under sequenced as compared to my reference sample. Does that make sense? Or are you saying it just because I repeated this? Yeah, you're finding the median. Yeah. So you are finding the median, saying that, okay, if I'm comparing all the genes to a pseudo reference, right? Right? That pseudo reference is a measure of overall, given there were multiple samples, how over or under expressed is my gene with respect to that particular pseudo sample, reference sample, right? Now, if nothing had changed, what would this number be close enough to? 1, right? Like in some sense, some sort of 1, right? It won't be exactly similar because, these are geometric means and so on, but, you can expect that by some scaling factor, both these factors which have been sequenced are going to be similar, right? Because everywhere you see that this number is close enough to 1. In this case, when will the number not be close to 1? Is here I see that, okay, this is, close enough to 1. The reason, if this sample was deeply sequenced, which means all these numbers are going to be higher. They will, if this was sequenced 10 times higher, all of them will have a overblown factor by 10, right? When you calculate the geometric mean ratio. That is a number that you're trying to estimate by figuring out that if I compare this with a reference sample, what is overall, by how much is my each gene scaled by, and that becomes your size factor. That's what you're trying to calculate. Does not? Why? Yeah. Hmm. Why does it make sense, right? So very good question. The reason your median is a good interpreter in this case is it's again, what is your overall assumption? That 50% of the genes will not show any change, right? Which means that they're not differential. But they will still have that scaling factor, which means they will still be high by a factor of 10. But they're not going to influence this calculation, which is right now influenced by one very large number. So that's where median is useful. You could not calculate the average of this because, see, for this example, right, it's clearly influenced by this value here. So if you calculated the average, it will go ORI, right? It'll go 2.33 by whatever it is. 2.33 is like 0.7, and this will be whatever 2 factor. So it's extremely going to be influenced by one outlier gene, which you're avoiding by calculating the median, because that median captures that most genes are not undergoing a change. If the say that again, sorry. Yeah. The median could be anything, right? Like at this point, what you're trying to say is, with respect to overall, all the entire samples, I don't know, like I know their total disk depth, right? So I have some idea that maybe this was sequenced by 10x factor, right? I can guess it. But when you calculate this pseudo reference sample, you are trying to bring all of this to one level, and then you're comparing this with this reference sample. This overall ratio that you get, right? Ideally, it would give you all identical numbers because they have all been scaled by the same number. But in this case, the reason you work with median is to tell you that some genes can change also. So be ready for that. So don't calculate mean. No, not from 1. 1 is an example I gave, right? But this number could be anything. Like this number, right? I chose a very simple example, but it could be anything. It could be like a factor of 10. This just depends on like what are your counts. It just tells you, you'll do this exercise and you'll see what happens. But this number could be anything, like 10, 15, 20, whatever. Or even less than 1, right? Yeah. Yeah. Mm-hmm. Yes. 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 This fails. It won't work. This will not work if 50% of the genes go weird, this will fail. Normalization will not work. And we will go to that case when we start to talk about single cell. The same, so stay with this idea, right? We are working with bulk. The moment we move to single cell, this assumption will start to fail, and we will not be able to use exactly the same idea in that setting. We'll start to cover this in like a couple of sessions. Okay, but everyone understand this is very crucial. This gives you a lot of hold of your data, like how to handle data, how to sort of think about overall analysis. So we've exceeded 3 minutes, so I will stop here. I've given examples here, but I really want to, yeah. So once we come back, right, and finally in the next session, hopefully we'll finish the hands-on. I'll come prepared, and I'll also ask you to come prepared. We will talk about like why are we doing all this and how to actually think about the distributions in general, how to model this kind of data. We are not done yet, right? So far, we have just normalized things. But if you remember, what we wanted to find out is which gene is differentially expressed. So next class, when we come back, we will talk about models to capture this and how to model that kind of information. Okay. Thank you.